Good afternoon, and thanks for having me here. It's always feel good to be back home. I call it home here for myself. So thanks for your time, for attending the talk today. So, so this work that I'm going to present today actually is from my postdoc research that we published actually this year. So it's predominantly looking at the subthalamic nuclear activity during speech production versus limb movement in patients with Parkinson's disease, that from now on I'm going to call it PD, so just the jargon that we use a lot. So what is Parkinson's disease itself? Parkinson's disease, first described by James Parkinson, he called in his lecture, like in his essay, as a shaking palsy. It means that the patient shows predominant symptoms is the tremor at rest. So then later we realized, learned that it's a progressive neurodegenerative disease that is caused by the death of dopaminergic neurons in the basal ganglia, particularly in substantial nigra, that can also affect the different cortical region through different pathway, hyperdirective and directive pathway that they have at the basal ganglia. Currently, more than 1 million people in the US are suffering from Parkinson's disease. And every year we have about, a new study came out in 2022 showing that every year we have 90,000 new cases of Parkinson's disease, with the prevalence that predicted to be around 1.3 million by 2030. So this is the basic symptomology of Parkinson's disease. So what is happening in Parkinson's? Can you see my cursor?
Yes.
So usually this is adopted from the Nether's neurology. So okay, it's right here. So in PD usually starts with slow progression. Usually it's a tremor in upper limb. Usually started on one side, and patients usually show tremor at rest, but when they're doing active task, that tremor goes away usually. So it's not during the performing any motor task. The other main symptoms of the Parkinson's disease is bradykinesia, which the patient usually show slow movement, and also they have a hard time to coordinate their movement to perform that in a smooth manner, such as tying their shoes, or doing some fine motor task, and it takes longer for them. So at the bottom here, you see the different stages of the Parkinson's disease. It's based on the Hohenn-Yahar scale of Parkinson's disease. It's a 5 scales, basically. So in the stage 1, it's usually unilateral. Patients are completely independent. They can drive, they can do what they used to, they can handle their careers. In stage 2, it becomes more bilateral, and with early postural changes that they use to have a forward postures. It's a really unique feature of Parkinson's disease. When they're walking, they're more forward posture. And then after, in the stage 3, we see pronounced deficits in the gait. It means that they have a hard time to start walking and handling their issues in everyday life. And in stage 4 and 5, usually patients show significant decline in their independence. Basically, they rely on other people, and at the end, they're going to end to the wheelchair and die from Parkinson's disease. The whole course of Parkinson's disease can last from 10 to 20 years. Some patients show slow progression, some patients show fast progression. But what are the neural markers of the Parkinson's disease, as we know, in terms of the neural oscillatory markers of the Parkinson's disease? It is basically the exaggerated or abnormal activity within beta band, 13 to 30 hertz in cortical and subcortical structures. Sometimes it refers as a beta park, basically. This is in the top panel here, in the panel B, you see a exaggerated increase in the power of beta band in the primate model of the Parkinson's disease that they induced in the laboratory. And this is recording from the human subject from the STN. subthalamic nuclei that you see like exaggerated oscillation prior to the onset of the movement. And we do like the similar things, collect the data non-invasively using MEG. You will see like, usually we see decrease in the power of the beta oscillation, as you see my cursor here, when they start the movement. When they are about to start movement, you see significant decrease in the power of, right here, and you have like a rebound after, basically. Then the time zero is the onset of the movement. But in patients, you see there's a decrease in the beta band desynchronization, abnormal beta band activity in their patients. And they have a like less prominent beta rebound after the movement onset, showing that the beta band is the abnormal beta band activity in cortical regions and subcortical regions are the neural marker of the Parkinson's disease. So when we come like looking at the Parkinson's disease, the course of the disease and the progression, most of patients, more than like 95% of patients actually suffer from some speech and voice disorders. And it significantly affects their functional communication and everyday life experience, communicating with other people. So Parkinson's disease speech problems are like characterized under hyperkinetic dysarthria, which means that these patients have like a decrease in the range of the motion in their articulators. So as a result, they show hypophonia, which refers to like decrease in the speech volume. So they cannot speak loud because they cannot get enough contact or closure in their vocal fold to create enough pressure under the vocal fold and create like a loud speech. And you can see that an example of that hypophonia in this slide over here in this image. The top is like asking patient to repeat 15 times the word P, and what you see over time, this is the amplitude or intensity of their voice. Over time, after repeating, their amplitude is decreasing. It's the sign of like hypophonia in these patients. The similar pattern, if you look at their spectrogram, looking at the first format, it's almost there. The second format is decreasing after a few repetition, and you don't have like a third format even in during their repetition. So these are like a psychoacoustic features of hypophonia. Then patient may also show misarticulation because the range of the motion is decreased. They cannot hit the right target when they're trying to produce the speech. And as a result, sometimes they become faster and slower than other people. And they can show both slow speech or like fast speech, which make them disfluent. And altogether, these like deficits makes patients with Parkinson's disease less intelligible for other people, like for their like communication partners. So then the interesting question is that what are the neural markers when we look at the speech deficits in Parkinson's disease? This is a brand new paper actually published 2 weeks ago. It's showing like fMRI images like and the brain activation during vocalization in patients with Parkinson's disease versus control matched healthy subjects. And what they found is that the patients with Parkinson's disease have hypoactivation in the dorsal premotor cortex, and interestingly it wasn't more in the right hemisphere than the left hemisphere. And then we want to look at other parameters like from the electrophysiology, from the EEG, from the work that I did here during my PhD actually. We looked at some EEG activities correlated with associated with the speech production. We looked at the premotor activities and postmotor activities. On the top, this here is for controls, and the bottom is for PD. And you see, like around the premotor, like pre-movement, before the onset of the speech, in Parkinson patients, you see significant decline compared to the controls in the amplitude of the EEGs. And when we do like take all together all 64 channels that we collected data, and the time zero here in the is the onset of the speech. They see Q on the screen, and when they see the Q, they're gonna say like, ah, just vocalize. And you see like a significant reduction in the amplitude of the global field power, which is corresponding to the average of all, like an average of standard deviation of the all EG activities, ERP activities, compared to the controls. And this is like we call, like sometimes people refer this as like readiness potentials, and we see that significantly decreased prior to the onset of the movement and also during execution of the speech sounds itself. So what are the treatments for general motor symptoms in PD and how they affect speech and limb movement? So that's a interesting topic itself. But usually as a first line of treatment, they get like dopaminergic medications, such as L-Dopa, to improve their motor and non-motor symptoms. And they show significant improvement in early stages, let's say like 3-4 years. But after, over time, dopaminergic medicine becomes less effective and induce some side effects, such as dyskinesia, involuntary movements in their gait and in their talking, in their walking. And there are like some evidences there, like from a follow-up study, like the 2 years follow-up study, a recent study, showing that after 3 to 6 years treatment from the L-Dopa, you actually see neurogenic stuttering, actually. Patients start showing stuttering symptoms as a result. So then it's the time for patients to get a new type of treatments usually, if the patient don't show significant improvement and suffering from side effects. So that line of treatment is the deep brain stimulation, basically. So the deep brain stimulation is a very promising method. It's invasive. What they do, they implant electrodes in the deep structure in the brain, such as subthalamic nucleus, and it goes through the skull. It's a small burr hole in the skull. They open it, and they implant the electrodes in the deep structures such as STN and GPI, and it has like an amplifier outside right now in the patients, and the neurologist can control all parameters for stimulation, basically. And these days, even these turn to be like an active amplifier. They call it brain sense. So basically, you can unlimitedly over 24 hours, 7 days, you can collect directly data from the STN and those parts. So this is adopted from the Medtronic company. I don't have any association with them, but we work with them on with the data acquisition and other things in the operation room. So this is the schematic view of where is the STN actually. And these are like a DBS leads goes, and these showing like the electrical currents when you stimulate the STN and how it can reach to the surrounding neighborhoods. And actual deep brain stimulator itself looks like this. It's similar perhaps like things for like the electrical stimulation anywhere. You have like usually 4 contacts sitting in the STN. They implanted here. You usually have 4 contacts based on the patient's symptoms. They define the target and implant the electrodes basically there. And after surgery, usually neurosurgeon can flip and change these, which one going to be positive or which going to be negative, creating a dipole. They're very focal, but it's a matter of millimeters. It's usually 3 to 2 millimeters of the structure. And the whole goal is that here, to normalize abnormal oscillations in the STN with the hope that we can modulate the STN itself, motor symptoms. So if you look at the STN is located underneath of the thalamus, anatomically, and under the STN we have substantial nigra that it's usually used for other type of treatments. Like I saw there literature showing that the substantial nigra is for gait issues in Parkinson's disease. If they have more gait issues, they implant electrodes here. But usually the target is the dorsal portion of the STN, which is the motor STN we call it. And they try to implant electrodes here to modulate directive and hyperdirective pathway with the motor and premotor cortex, so they can suppress abnormal activity and improve patients' main symptoms. So here's the real time experiment in the OR usually happens. So in here, in the top panel, you see a time frequency panel activities for the STN activities in a patient, in just one patient, and up to 30-35 seconds, They did high frequency deep brain stimulation, which is like around like 130 hertz to like 180 hertz. And do it like single pulse, you see like immediately causal changes. I mean, the tremors really stops if you are in the right target. So, what's happened at the bottom, you see, this is the amplitude of the movement. So when you have these suppression of blue colors here, I show in the cursor, you see increase in the amplitude of the movement. Means that they increase their range of the motion. They can move their hand, or like if you ask them to tap their finger like this, at the beginning is like this, you stimulate it, immediately increase in the range of the motion. So it's really causal, in a matter of milliseconds, you're going to see the effect. But once you start the stimulation itself, you see exaggeration occurring. I mean, over here, and the movement amplitude is going down, means that it's decreasing. So it's a real like a causal effect. I mean. So the other thing is that you can do the same thing in the GPI, the globus pallidus. For the patient that usually have more severe cognitive decline, they do. It's not a very common target for deep brain stimulation. So they usually do it for the patient that they are having like a low or like a low score in like neuropsychological tests usually. So what's happening here, that it's time zero, it's when they turn off the high frequency deep brain stimulation. You see this is blue all the way here, and it's like suppressing the beta band activity basically, exaggerated beta band activity. And once you stop it, you see it's occurring again. I mean, it's becoming more red, and it means that they are like more exaggerated or like synchrony within the STN. And as they showed in this slide, like the results, this is like the amplitude of the movement, and this is like the beta band power. The patients with the higher amplitude, like higher beta band, like a more red, I mean, like an increase in the power of beta band, is correlated with decrease in the range of the motion. And if you suppress that, you get larger like amplitude, like larger range of motion basically. So, what are the side effects? Then we talked about side effects of levodopa. Then what is the side effects of the DBS STN basically? For limb movement, for majority part, there is no like significant decline. Actually, you see significant improvements in the limb motor itself. Like tremor goes away, they walk, they can go even play golf after it. So the patient had lots of tremor, they go and play their golf, they're fine. But they are like about speech and cognition and the language itself, I mean like verbal fluency, you see like inconsistent results. So everything is variable and varies from patient to patient. And actually, as like a matter of like a functional communication, when I talk to someone, it's important things they understand me. The speech become more intelligible. But these patients, a year after surgery, showing like about 14 to 17% decline in their speech intelligibility, which is a significant decline. And also, other aspect of their dysarthria can be also exacerbated by the deep brain stimulation itself. So the question comes to my mind, okay, why STN DBS responds differently to speech versus limb movement? And we had like initial hypothesis about it when I was a postdoc in Iowa, and I'm going to explain what is the hypothesis itself later, but this was the question came to our mind, I mean, actually about this. So then we looked at, okay, let's look at the STN. We have this opportunity in the OR, actually. I'm going to play a video of the OR, operation room, that it's awake surgery, the deep brain stimulation surgery. It takes about 4 hours, but the patient is completely aware. They get the local anesthesia for their skull. And what is happening is that, let's see, I play from here.
When you bring the left hand up, touch those fingers together, almost. Okay? Now, go ahead and relax the left hand and bring the right hand up as if you're eating. Very good. Bring it down your side. Relax.
So what is happening here is that this is a normal procedure for deep brain stimulation. Neurosurgeon usually start implanting, like putting like 3 temporary electrodes to detect the target for the patient based on the neurophysiology in real time, basically.
Make sure it's keep speaking this way, people are asking, sometimes the sound cuts off.
Okay.
The microphone is not doing it, it's your laptop.
Oh, my laptop?
Yeah.
Oh, sorry. So the, I start talking louder. So, what is happening in the OR is that the neurosurgeon usually use like 3 electrodes, like these are high impedance. The tip of the electrode is about 1 millimeter, like that's literally like very tiny. And they drive it through the burr hole to get to the predefined target using neuroimaging. And once they get there, they start asking patients to repeat certain tasks. I mean, like tap, or like do like pedaling, I mean, or like they ask open or close your like eyes, things like that, like a motor task, so they can see real-time modulation in spike activity or neural activity in the STN. And when you get to the motor STN and they're moving the electrodes down, you will, expert neurosurgeon even can detect the STN from the noise of the firing rates of the neurons. I mean, so, and they usually look at the something like this, showing like 3 electrodes that usually they use it in the anterior. Okay, this is usually anterior, middle, and posterior STN borders. In the dorsal STN, they implant those, and you see these like tiny spikes coming out in the plot. I mean, here. These are like cell responses to the movement, basically. They're modulating by the movement. And you can see even like a PCA, principal component analysis, in real time. And these are like a waveform for the like single units. And based on these, they define the right target and get the parameters for that and implant the deep brain stimulator itself. So basically, during this time, they try to find a target. But as a research purpose, we have this opportunity to halt surgery for 15 minutes. And IRB doesn't allow more than that because it's a human subject, right? So we do like 10 to 15 minutes of research during the surgery. And what we do, we ask patients to perform certain tasks and we look at how the neurons are getting modulated. There are lots of literature about the modulation of the neurons in the STN actually. So like this one is in this panel. Okay, so this panel is here. It's for the spike activity as a firing rate in frequencies and in hertz for just reaching tasks, just trying to reach to something in the OR. They show the cup, okay, try to reach to that cup, I mean, for example. And you see these like tiny dots here, like each line here is representing a trial, as you see here, and every dot here, it's a one spike, basically. So it's like a neuron is discharging in response to a movement, and they are showing in a way over a period of time, usually like 1 second, 2 seconds, like the whole course of the time. And the same thing if you ask a patient to like just pedal or like do heel strike, you see like interesting pattern of firing of neurons. I mean here, like going up, there's no like tapping with the feet or heel strike, and there's another one, and you see the modulation increasing in the firing rates basically. And these like, as I said, these black dots here, I mean, each of them are like one discharge for the neuron. There are studies about the speech and STN, also like how neurons respond to the speech in STN. This is a study showing that you're asking patients to just like repeat some speech sounds. I mean. So, and in the panel A, it shows like spike activities for 5 speech sounds. And the red line over there, it's a time for Q. They hear a cue, and in the blue line that you see dashed blue line in the panels, right here, they are starting producing the speech sound. From here to here, you see increase in the firing rates or number of spikes per second, we define it. And you see like a number of spikes in the raster plots up here. You don't see the same modulation during in the subpanel B. You don't see similar modulation during perceiving the speech sound. So it's only during the speech production. And as a control condition, you don't see any response in the panel C when they hear like a beep sound or like a cue sound, I mean, that be ready to repeat the task. There is not that modulation. In other recent work, I mean, in this panel I'm showing from Lipsky et al., they found like 3 patterns of the neural activity in the spike activities. during reading words aloud, and they found neurons that they are showing decrease in the firing rate, increase in the firing rate in panel A, decrease in the firing rate before the onset of speech, and also showed like a mixed pattern, decrease, increase, and then decrease during speech production. So they find like heterogeneous pattern for the speech production units. So we have like this literature about speech production and about the limb movement. The time that we studied, and as of today I am not aware of any study, there might be some, but I'm not aware that we didn't have any study directly comparing STN activities for speech versus limb movement. And one of our initial hypotheses was that one of the reasons why speech results following deep brain stimulation is inconsistent. It might be because actually STN neurons are like differently encode speech versus limb movement. So we did like a very simple task during awake brain surgery, deep brain stimulation in 12 PD patients that they went under bilateral deep brain stimulation implantation. We collected data while they had like 3 microelectrodes in right and left hemisphere. And we asked them to perform simple tasks, either like repeat single syllable, like ta 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 ta, or vocalize and say ah and hold it for a few seconds, or tap, just for like 3 seconds, just like tap with their finger. And we collected data and synchronized their timing with the neural data, so we can extract time-related activities within the STN. So for our analysis, this means partly done with my colleagues actually. I performed like some of other like analysis for this part, but we extracted the data, and first we bandpass filtered from 2000 to 6000 hertz. Usually bandpass filtering for spike analysis is between 300 to 600 hertz, 6000 hertz. But we start with 2000 hertz. I'm going to explain why in the next slide. After high-pass filtering, we looked at the data and removed any OR-related noises. Basically, there are lots of like devices in the OR, like creating lots of noises and artifacts in the data. And also, patients have tremor, so if there are like any tremor-related activities, we remove them from the data. Then we used thresholding at minus and +3 standard deviation to detect spikes using Telexon software, which is a commercial software, and we manually isolated the single neurons based on the principal component analysis and how they look in the PCA environment. And as you see, that's like the distribution of the, and the plot I showed, this is the distribution of the waveforms, and this is the mean waveform that they detected by the PCA as like a single unit activity. I mean, so in the STA, just nothing about the speech or limb movements, is just like pure STN activity that we isolated single unit activity in the STN. And on the right side, you see something called like spike intervals that shows the intervals between each spikes. So single units cannot fire every 10 milliseconds. There is a refractory period for the action potentials that usually it's like more than 10 milliseconds. I mean, so if it's less than 10 milliseconds, it's less likely to be like a single neural activity. It's probably coming from multi-unit activities that we will also discuss about in my results section. So we also like aware of the studies that looking at the STN, seeing that there were like both single units and multi-unit activities within the STN. So we decided to perform additional analysis using mountain sort toolbox to detect our multi-unit analysis. With multi-unit, we mean that not a single but population of the neurons. They can be like more than one neuron, they can be 10 neurons, 100 neurons, but they are still local. So they are located in the STN, and the activity will reflect the activity within the STN. So this is the main reason why we didn't, why we did filter our data from 600 to, from 2000 to 6000. we listened and we came with this like really nice unit you see here. This paper actually published in Neuroimage with my colleague, last year. So you see like beautiful like a histogram of the like a single, like I'm gonna say that this is beautiful multi-unit activity, and you're gonna feel that this is like genuine and you're gonna believe in it. But we came across and we plotted spectrogram for this, and you see like there are like four-band type responses that you will see. And these are actually chasing or following the fundamental frequency of the subject voice in microelectrodes. Basically, when they're vocalizing, it's vibrating when we call it microphonic effects. And when you do like cross-correlation, it's highly correlated. The time of onset of the voice is highly correlated with spike amplitude, actually. And if I play that sound here, this is the waveform of a neural spikes that we recorded. We convert it to audio file, just for our curiosity. You're gonna hear some like ah and tahs in the background. Let's see. I didn't test that one. Sorry. I tested the audio, is working fine or not. I didn't test that part. But you see that you can clearly hear, that's literally, if you plot it like a wave and look at the wave itself, you're going to say that this is really like beautiful, like action potential, like happening there. But literally microphonic effects. What we did, we applied 2000 hertz as like our high pass filter. We get rid of those. We lost some genuine neural activities that were contaminated, but we still were able to detect some single and multi-units actually. Okay, so what we measured, so as our measures, we measured two indexes. One of them was like absolute firing rates, means that the number of spikes per second with isolated neuron in the PCA, or like by the mounted sort, and firing rates of during a specific task, like speech and limb movement. So it was the whole entire trial, across trial, we calculated it and we got it like as a firing rate in hertz. Then we had another measure that we used, we call it relative modulation index, which is more sensitive than the absolute firing rate, and it can capture transient changes in the action potential around the onset and offset of the tasks, basically. So, and this was defined by the firing rate of the trial minus the firing rate of baseline divided by sum of the firing rates of interval and baseline. And if this number comes positive, it means that there is an increase in the firing rates around the onset or offset of the task. And it becomes negative, it means that there is a decrease in the modulation of neural activities in the STN, basically. So I'm showing like some single unit activities actually here, and then I will show like some multi-unit activities. Here we see like responsive units for speech onset. Time zero here refers to the onset of the speech, and anything before that is the baseline. There is like real activity. And after that, you see increase in the firing rates at this plot. And these are all like these like small dots here, as I said, these are like spikes occurring over time and in each trial, so, across the trials. This shows like increase in the firing rate following the speech onset. But we have another unit that we isolated. You don't see any changes over time relative to the baseline, and we have actually less spikes. So this unit, we called it, it's not modulated by the speech task. For the limb movement, the same way, I'm showing here like example of responsive units. You see relative increase in the firing rates. This is the baseline, this is the onset of the tapping, and you see slight increase in the firing rates here to like 30 hertz. And these dots, like red dots here, shows the end of the trial. So there was like a difference from trial to trial, how long they tap. And in this panel, you see there's no difference between baseline and the actual and the trial response. Actually, the limb movement response itself. There's no difference between the firing rate here and the during task. So, and when we look at the multi-unit activities that we isolated, in the panel A, you see exemplar unit that responded to the speech, increase in the firing rates, and increased significantly. The same unit, multi-unit, didn't show significant difference, slight decrease, but not that different from the baseline for the limb onset. In the panel B, you see an exemplar unit for the limb movement. There is no difference here for the speech, but here for the limb movement, you see a slight increase in the firing rates from the baseline. And for the offsets of the task, we don't see that much modulation in these two exemplar units, basically. So what we did we got very heterogeneous pattern of neural activity. It's really hard to characterize them, but we came with the idea that, let's say, look at each units as their, and compare each unit RMIs across the trials and test them against the null value of 0, if they're different from the 0 or not. And we got about 69 units total we isolated. And from those 69 units, we found like 17 single units and 23 multi-units. From those, 26 got modulated only to speech. So they only responded to speech. 12 were like single units and 14 were like multi-units. We had the limb-only neurons that they responded for tapping only. They didn't show any activities for speech. There were like 3 of like single units and 7 of multi-units. We had like about 4 units, 2 single and 2 multi-units, that they responded for both speech and limb movement. And overall, we had like 29 neurons that they didn't get modulated with any specific task. Then we look at the pattern of the increase or decrease in the firing rates. We see that in both speech onset and offset, we see increase in firing rates, about 15 and 10, and therefore limb is like 7 and 1 unit. And we had 5 and 3 units show decrease in the modulation index, basically, or RMIs. And for limb, we have 3 and 3, basically, for onset and offset. Basically, we find very diverse pattern of firing rates. Some units only modulated by speech, and some units modulated by limb, and few units modulated both tasks. So when you want to like run stat on these data, it can be problematic because there's lots of variance in the data. But even regardless of that, we performed some statistical analysis using LME. So what we found actually overall, it becomes significant. Overall speech firing rates, and their modulation index increases, versus limb, it showed decreases overall if you want to compare these two tasks together. But when you look at their absolute firing rates here, it's both of them showing like an average of firing rates about like 40 hertz for both single and multi-units. We didn't separate single and multi-unit activity for our statistical analysis because when we tested them together, there were no difference. Like including unit type as a single and multi-unit didn't show any interaction actually with the task.
Is this absolute firing rate after the task starts, or
Yes.
just before the bit? So after.
Yeah, yes. But the modulation index, it's relative to the baseline also. So for the conclusion of this part, I think I'm good at time. So STN neurons, we showed that the STN neurons were differently modulated by speech and limb movement. And they are like our hypothesis and some direction for future studies actually. So we think that in general, DBS implantation and configuration are biased toward improving main symptoms, which is tremor and their gait tissue. High frequency deep brain stimulation improve motor symptoms in the PD. But we think that it interferes with the neurons that are related to speech, because during the surgery, they are targeting the neurons and find the right target based on the limb motor symptoms most of the time, and they're ignoring the speech side. And as like a further step that is like more like as a like initial hypothesis for our team is that we think that DBS alerts that local activities, but also changes the connectivity between STN through hyperdirective pathway with the IFG and as well as the premotor cortex, causing like some post-deep brain stimulation or deep brain stimulation induced speech deficits after the surgery. So this is like all like an initial hypothesis, but we're still working on it. And as you see, like in this study, there are like some recent evidence showing that there is a spatial map between STN and IFG and the premotor cortex and different parts. And actually, what happened in this study, they stimulated the STN and they got the potential in IFG basically. They immediately changed it there. So what is the next step in our research actually that I collaborate with our colleagues at University of Iowa? We want to do something like this that we started. So it's multi-regional recording. So it means that we are recording activities from the STN, different part of STN, and we have opportunity to slide some strip electrodes temporarily during surgery in the premotor motor cortex, prefrontal cortex, and we can collect simultaneously within the same patients the task like we collected during the sentence production or like syllable repetition and finger tapping, and this data is still like being collected at the University of Iowa. So the whole goal of this is to just understand how STN and subcortical structure contribute to the speech production of itself. So I want to thank my colleagues, Dr. Greenlee in the University of Iowa. He performed the surgeries, and my colleague Joel Berger, that he is expert in the neurophysiology and helped with the analysis, and other collaborators that helped with the patient recruitment and also contributed to other part of the research. And as a matter, I am co-editing this specific research topic in the Frontiers that is about the Parkinson disease. If you are like working on the area of the Parkinson disease and speech production, we encourage you to submit your papers. It's open, and we are looking forward to that. Okay, and thanks. And any questions?
Thank you so much, Karim. Let's see if we have questions from the room first. I think it's probably a good idea for the audience online if you repeat the
Can I see the questions here or no?
You can, but the first gonna do the questions from the room.
I don't see it. I need to end my presentation.
But I'll read out the questions.
Okay.
So, is the problem, you mentioned, so I guess, understanding the problem in the implantation is that the surgeons are basically looking for limb responsivity to the stimulation, but not speech. So is that simply fixed by having both tasks and then just having the surgeon to optimize the location based
So actually,
You're gonna have to repeat the question.
okay. So the question was that it's the problem fixed during the surgery if they tapped and talked and recognized the same units. So basically, we didn't fix that problem. That's the question, but that's the plan. So we try to understand. That was our hypothesis that there is a difference in the units. So we found it. So there is one hypothesis that since it's a high frequency stimulation, it impairs speech but improves limb movement. But there are evidence that if you do low frequency stimulation, let's say 4 hertz stimulation, you implant everything, go out, and the patient comes after a month for the neurologist to set the parameters. There are some evidence that if you put on the 4 hertz, 10 hertz, it shows both improvement in the gait and the speech.
So thank you so much for this presentation. It's great work and definitely needs to be done. I've seen a number of patients post DBS who are walking and moving just fine but have complaints of voice and swallowing problems still. And so I was looking at it just with a little bit of a different lens, and I think the acoustic signal that you showed of the P repetitions got me thinking a bit of patients I've worked with Parkinson's disease and the co-occurring respiratory function disorders that happen and how with limb movements, that's pretty a one-shot thing, but with speech, you actually have the co-coordination of the respiratory system and the speech, which makes it a little bit more complex, especially when you're separating out neurons and the STN and that sort of thing. So how do y'all think about and kind of devolve that?
Yeah, so the question is that the actual question was that it's hard to put like summarize, but
Yes.
I can summarize it.
Respiratory, how do you consider the respiratory system in your
So
data?
about like speech, the question is that speech is more complex. It requires activation of different systems, respiratory system and laryngeal system, and you have articulators all together, versus limb movement, it's one direct shot.
Right.
And how we think about it basically.
And especially with the respiratory system, since that co-declines in
Yeah, yeah.
Parkinson's disease.
So yeah, so for the respiratory system also, like I think that's a good question, and I have like a speculation about it. I don't have any solid evidence for that. So actually, for most part of my PhD, I was like comparing speech versus limb movement, and one of the like initial thought was that the speech is more complex. It requires activation, coordination, like several systems, as you mentioned, and the respiratory is one of them actually, and it's an important one actually. But in Parkinson, as you mentioned, like you will see like decrease in the activation and how the muscle works in the respiratory system, and it's going to affect that. Actually, as a matter right now in University of Iowa, we have a colleague that he's collecting breathing data during intraoperative recording, putting like a belt in the patient that's going through surgery, and simultaneously collecting data. That's another interesting part that they're looking at.
Yeah.
Yeah.
We have a question online from Enriqueta Canseco Gonzalez. She asks, do the spikes depend on whether the patient is speaking in response to an instruction versus speaking spontaneously?
That's a good question. I don't know, but we could look at it actually. We can go back and look at our data. We never analyzed actually, so usually neurosurgeon just like randomly chat with the patient to keep them awake. So we can look at those data, patient responds, and they get to like a spontaneous type of speech, and so we can compare that, but I don't have any answer for that.
Okay.
I have a more of a methods question because I'm not as familiar with this, but you had indicated that you kind of filter anything that's less than 3 standard deviations for the spike analysis. And like in our data, we're usually like excluding people who are outside of 3 standard deviations as outliers. So I was just wondering if you could explain why you set that threshold and you ignore sort of what's underneath that.
Yeah, so the question is that why we include like detect our spike activity above 3 and below 3 standard deviation. So spike activities, it goes the other way usually, like the other like a normal data analysis or like behavioral analysis that usually keep them as an outlier. So usually you have a background activity that is like in the resting state, there is nothing, and suddenly something is happening, and it passes like 3 standard deviations. So neurons can be like a discharge like a both way, and it's going to be positive and negative on the discharge. So that's why you see like both spikes like should pass certain threshold to be detected by algorithm.
And is there any reason why it's set at 3 and say not 2? Like, is there something that you may be missing?
Actually, so the question is, oh, sorry, you have extra thing to ask. Okay, so the question is that why we pick like 3 standard deviation for the spike detection. So spike detection usually it's based on like the classic neurophysiological working, even in the experimental animals actually. They do the same thing if you do manual spike detection. Usually you apply like 3 standard deviation. But it can be like a more conservative. It's gonna go like you can make it like even 4. You can make it 5. I mean, but what I saw in the literature, it's like usually 3 standard deviation.
We have 2 questions in one. I had a question. Are there any inherent differences between the type of units that respond either to speech, limb movements, or both? So in other words, what do you think underlies that differentiation Can it be the fine-grainedness of the type of movement, for example? And related to that, I think, is a question from Heather Kabakov. It's fascinating that STN neurons differentially encode limb versus speech movement. Do you think that it goes even further, saying that STN neurons differentially encode different aspects of speech production, say pitch versus formants? Patient speech outcomes are so variable, so STN location may further play a role?
So the first question is require more deep analysis in the data. But I know as a matter of recording, location of the recording does matter in this scheme. If it's anterior, middle, or posterior. And even like if you implant electrodes in the anterior STN, usually you get better speech outcome, so versus other location in the STN. So, but regarding the like type of the neurons how they were, we didn't do further analysis for that. But I think that they're like inherent different, they're like some like variability there that we couldn't control. Some of our results may be biased with that actually. There
And
wasn't
then-
the second part of that
Yeah.
question, sorry.
So the different, maybe, is it possible that STN neurons differentially encode different aspects of speech production, so that there may be neurons that respond more to pitch changes and others to format, or...
Yeah, so STN even like differently, we are like having like some tests that we're collecting right now as like maybe 5 or 6 patients that I can tell. We see like different, even like a syllable repetition versus sentence repetition, more complex multitask. We see the difference in modulation. And I think that there's another aspect that we published in the paper and discussed is that how you know that it's induced by speech versus like a articulated movements. And actually, we're collecting some data that show, and we're going to publish soon, it's that there are like some differences between just like if you open and close your jaw, take your tongue out, or like just move your tongue around, and versus when you're like producing actual speech. There are like different nodes responding differently.
Okay, so that comes to me as a follow-up question is why should that be the difference? Like the theory of what's being encoded, that it's not purely the motor effector velocities or positions, right? But I'm curious what that means if the same effector is differently encoded for speech versus non-speech. But my other question I was going to ask is, in some sense, it's not surprising to see the differentiation because they are different effectors, right? Like if you're moving your arms versus moving your vocal tract, those are different muscles. They have different targeted fibers, right, by the end. But it was interesting to me that there actually are neurons or single units that were encoding both and what those are doing, because then it's no longer just coding a muscle contraction, right? It has to be something else. So those are my 2 questions. How can it possibly do speech versus non-speech with the same effector, but then what are the neurons doing that actually respond to
So
different
for the first one, so like here's the thing. So STN.
You're going to need to repeat the question.
Oh, sorry. Those are like 2 complicated questions now. Can you?
We need
Okay.
a
The first
microphone.
question was that actually it's a good question. I want to like chat about it. Actually. So, the first question was that okay, having like speech and non-speech. Okay, and what is the difference there? Why you have like different units from like separate tasks?
If there's a single neuron, if there are different, so you can move the same effector,
Okay.
but it will differently modulate the neurons depending whether it's speech or non-speech. Why? What's the theory of what the neurons are doing? And then the second question is sort of the inverse. What is it possibly doing a neuron that responds to both speech and limb movement?
So yeah, the first question is that about speech versus non-speech basically. And I think that if you look at the speech production itself, it's not just purely moving the limb around. There are some other cognitive mechanism going down. For example, in our tasks, patient needs to keep certain, like for sentence repetition, for example, they need to keep the sentence in their short-term memory, for example. It requires working memory. STN has a directive pathway with the prefrontal cortex published recently showing some activities between STN and prefrontal cortex during the working memory test. So there are like other mechanisms that involved in the speech production itself. I mean, depending on the task, you're going to see different recruitment of the neurons at the face, versus the just opening and closing, like the moving like simple articulators. And the second question was that, I forget it again, so sorry.
Speech and limb movements,
Okay.
there are some neurons that respond
Okay.
to both speech and limb.
So why those neurons are responding? Like there are like neurons, like about 4 or 5 neurons we found modulated and responding different, like similar to speech and limb movement. So that's a good question. We had only 4 units, but still, I think that it comes back to the similarity between two modalities, and can be, I mean, like in terms of the, even like in cortical levels, if you do something with your hand and you start talking, you're going to see some overlap in cortical levels as well. I mean, so I'm just speculating about it, I mean, so I don't have any solid response for that. But even in IFG, if you try to grab something, your IFG is going to get activated for grasping them. And when you produce something, it's your IFG. That's from what I read and what I know of it. But in the STN, it's plausible that we have some neurons that they are like responding for both of them.
But what are they doing?
They're just like there. I don't know exactly. Yeah.
I think that's a good moment to end the session. Thank you
Okay.
so much, Karim.
Thanks.
Thank you.